Good afternoon. Welcome back to Virology. 5 lectures left. Aren't you thrilled? Today we're going to talk about antivirals, antiviral drugs. Remember last time we talked about how vaccines can prevent disease, but once you're already infected, they really don't work, with one exception, which we said last time was rabies. In fact, you answered the question that it was rabies. But antiviral drugs can stop an infection once it's started, although you can't wait forever. You have to, especially in an acute infection, you have to get going and start treating. And that varies depending on the virus. So we've been looking for antiviral drugs. These, by the way, are prescription drugs. They are FDA approved. You can't go in a drugs, at least in the US, you can't go in a drugstore and just buy them. In many other countries you can, like antibiotics, you can buy everything over the counter, but in the US we regulate them. And we've been doing antiviral discovery for 60 years, but we don't have a lot of antiviral drugs. Only about 100 on the US market. And most of them are against HIV and hepatitis C virus and herpes viruses. What do they have in common? Do you know? What's in common among those 3 viruses? Folks, it's on the slide. You just have to read it. They cause persistent infections because they last your life, right? So we have plenty of time to treat you. So here's a graph of antiviral drug discovery. So on the y-axis, the number of approved drugs. Each line is a different virus. And you can see, when did this start? Back in the 60s, really slow until HIV discovery. Then it took off. Here, the HIV drugs, 41 as of this lecture. Probably it's even more. Then we have herpes simplex, hepatitis B virus, hep C is a big one, 18, and influenza virus. But there are a lot of other viruses we've talked about, right? What happened to them? Well, we'll get to that. Here's a nice chart, series of pie charts showing you drugs by virus and target. So on the left by virus, you can see again, most of the antivirals, 43 of them, against HIV-1, then HCV, hepatitis B virus, herpes viruses, influenza, and others. In the middle, what's the target of the drug? What does it inhibit? Most of them polymerase inhibitors. Then we have protease inhibitors, integrase inhibitors. All those are enzymes. That's the one thing those things have in common. They're enzymes. So there's not a huge amount of them around. They're catalytic. You don't need a huge amount of enzymes. And so it's a good drug target. Then NS5A is a hepatitis C virus protein. And then 10 of them are host targets. We have 10 antivirals that target the host. And that's shown on the last pie on the right. 75 antivirals hit the virus and 13 hit the host. There's discrepancies in the numbers, but don't worry about it because they're small. You get the point. All right, so why are there so few antivirals? Remember, viruses are locked up with the cell, right? They need a lot of cell functions. So it's hard to find a compound that interferes with virus growth, but does not interfere with the cell. If you do have such effects, you get side effects which are not acceptable. We can only take a certain level of side effects, and then we're not going to use the drug. And so every step in viral reproduction requires host functions, and therefore it's hard to separate the two. Another reason is that some viruses we can't grow in cell culture. Hepatitis B virus, human papillomavirus, very difficult to grow in cell culture, at least at the scale you need for drug discovery. Smallpox is dangerous, very hard to work with it. You have to work with it under a BSL-4 the highest level of containment. Yet we do have 2 antivirals which are stockpiled in the US. You know, we have a national strategic stockpile, okay, where there are vaccines and antivirals and other things in case bad things happen, which are going to happen in the next couple of years because we're trashing the public health system of the US. Just warning you, all right? It's going to happen. And that stockpile is used. The 2 smallpox antivirals are there in case of a bioterrorism attack using smallpox. Smallpox is the one virus that is a good bioterror weapon. You would just infect someone and have him walk into a major city, and then they could spread smallpox. And if you don't think there are people in the world that are willing to do that, I have a bridge to sell you. And Ebola and Lassa virus are also dangerous, so not easy to make antivirals against those. Third reason, most important reason why there are so few antivirals. A compound has to block virus replication completely. It has to be potent. And many of the drugs you take that are not antivirals aren't 100% effective. Aspirin doesn't have to inhibit its target 100%, probably 50%. I'm just throwing a number out there, and you feel better. But antivirals have to inhibit 100%. And if you don't, you're going to get resistance. So here on the right is a graph of viral load versus time and 3 different doses of an antiviral. The optimal dose, and this is viral load again. Optimal dose inhibits virus reproduction, but an intermediate or low dose do not, and the virus continues to replicate, and that will select for resistance. So you have to inhibit virus reproduction completely, and that makes it hard to discover antiviral drugs. Now, the other thing is, of course, acute infections are of short duration, or we should say defined duration. They don't last your lifetime like persistent infections do. But more importantly, by the time you feel ill, it's almost too late to take an antiviral. As you'll see, the influenza antivirals you have to take within 24 to 48 hours of feeling ill. In other words, feeling symptoms, right? If you wait, it's not going to have any effect. If we don't have any antivirals for polio, but if we did, you have a person who presents with paralytic disease, antiviral is useless. Virus is already gone by the time the paralysis happens. And so you have to give the antiviral early or prophylactically. So when we give the drug before someone is sick, we call it prophylaxis. Like PrEP, pre-exposure prophylaxis for HIV infection. But that's the only one we do that generally because it's not a good idea to give people who have no issues, no infections, a drug. But we do that for PrEP. And we don't have any broad spectrum antivirals. So if we did, if you had any kind of viral infection, we could give it to you, but we don't have them. We have them for bacteria, antimicrobials, but we don't have them for viruses. And we don't have diagnostic tests always. So it's like a chicken-egg thing. Nobody will make a diagnostic test because there's no antiviral. And nobody makes an antiviral because there's no diagnostic test, right? The companies that won't make any money. If there's an antiviral and no diagnostic test, they will never make any money. So only in certain situations do they work together, and it gets done, like a pandemic where you have everybody being infected. People say, obviously, we need diagnostic tests. If anything, just to tell you you should stay home, and then antivirals. Okay, a little bit of history. First searches occurred in the 50s. There were antibiotics of the class. This is called sulfonamide, and some of these seem to be active against viruses. So those were checked out. Synthesis of thiosemicarbazones were active against pox viruses. That's what this compound is here. And after World War II, smallpox was still a threat, so that's why that was ongoing. The 2 antivirals we have now were developed in the last 10 years solely for bioterrorism purposes because we eliminated smallpox in 1979. So there's very, very few people in the world are immune. And so if a bioterrorist released smallpox virus, it would be very bad. So we need antivirals and vaccines. And so, after around World War I, bacterial antibiotics were really effective. That's when they were first developed. And so the virologists said, maybe we can do something similar with antivirals. So for many years, companies did what's called blind screening. Now, companies have lots of chemicals. They have chemical collections that they make for various purposes. Thousands, depending on the size of the company, thousands and thousands of chemicals. And now you can buy them. You can go and buy chemical collections, and then you can see, does that collection contain something that will block virus reproduction? And so people just check them against different viruses. People also took soil samples, because soil has a lot of different microorganisms in them. And you can incubate it in broth and see if something is produced that inhibits virus reproduction. And then if you get, say, a broth from soil that inhibits your virus, you can try and purify the compound and see what it is and modify it to make it better, and then check it in animals and so forth. And you do things like reduce toxicity, increase solubility, bioavailability, and other pharmacokinetic properties that we'll talk a little bit about later. So that's called blind screening, because you're just looking to inhibit a virus. You're not targeting a specific mechanism. And you would screen thousands and thousands of molecules before anything got near people. One of them that was discovered in the 60s for influenza A virus is called Simitrel, or imantidine. It's a very strange compound, as you can see there. We didn't know how it worked until the 1990s, and I'm going to tell you how it works today. But we don't use it anymore because viruses are resistant. In many parts of the world, pig farmers fed it to their pigs so they wouldn't get sick, and that generated global resistance to the drug. So we don't use it, unfortunately. Today, the way we discover drugs is we use recombinant DNA, so we can produce a specific protein, a viral protein, and look for inhibitors of it. We know the reproduction cycles of most viruses, so we know what happens at every step. And even if you cannot propagate a virus in cells in culture, you can, say, make the RNA polymerase and look for inhibitors of it and use that. So we don't do blind screening anymore. We do very targeted discovery. And here's a generic reproduction cycle, which you should understand now, all the steps, entry and uncoding and macromolecular synthesis and so forth, release. We have inhibitors for every one of these steps: attachment, penetration and uncoding, mRNA synthesis, polyprotein processing by proteases, nucleic acid replication, that's a big one, integration, assembly, and even release. So the path to drug discovery is very long and convoluted. So you start with the medical need. Sometimes it's obvious. You have a pandemic, it's a medical need. You do some research, and you decide what your target is going to be. And when COVID started, we knew that coronaviruses encode a protease that's essential for virus reproduction. And so we had the target, and the proof of principle was that if you remove the protease, the virus will not replicate. And then you screen your libraries, your natural products, compound collections, combinatorial chemistry, RNAi, various screens, and then you get hits that are inhibitors. You modify them and eventually improve various properties. Will the compound get to the right place in the body? So there you have to go into animal models. Will the compound persist long enough to be effective? Pharmacokinetics. You can have a great inhibitor, but if you inject it in people and it has a half-life of an hour, it's useless, right? You can't be taking a drug multiple times a day. And will it be safe? Toxicity and specificity. So early on, these some of these things are looked at in cell culture, but eventually you have to do experiments in animals. And if you have a compound that makes it through all this, it goes into clinical testing, which means people. It has to be tested in people. And this is the discovery scheme here for a drug. So we've talked about discovery and synthesis. Preclinical is anything you do in an animal. And typically you need two. FDA often requires 2 different animal models for you to test your drug in. And then you do a phase 1 study, which is 20 to 80 healthy subjects. You give the drug, sometimes in different doses. And people volunteer for this. You may think it's scary that you would volunteer to be tested with a compound that's never been in people, but it's been in animals extensively, and we're confident that it's not going to kill people, and it doesn't. And it doesn't have horrible side effects usually. We've really studied it so hard that that could be eliminated. Okay, then phase 2, and look at the timing. We're already up to 10 years. It's taken us 10 years to get so far. Phase 2 is an efficacy between 1 and 300 people, and you see if the drug works against infection. And that means there have to be infections going on in the world, right? If there are 2 or 3, you can't do this. So right now, it would be hard to test new SARS-CoV-2 drugs because although there are infections and people die, they're scattered all over the place. It's very hard to get together 300 people who are going to be infected. But influenza is no problem because we have influenza throughout the winter all the time. And then finally, phase 3, you have more people, efficacy, side effects, and you have, if there's another drug already, you have to show this is better. If it's not, it will not be licensed. And so we're almost at 20 years. It gets all between these phases, by the way, the FDA reviews your data and says, okay, you can go to phase 2, you can go to phase 3. And then at the end, they license all of the data. They look at all the data and decide whether to license it or not. In other words, FDA licensure means the drug can be given for the purpose cited in the clinical trial. And then after it's released to the general public, we continue to monitor safety in many, many millions of people. And that's where you can get side effects that are 1 in a million because we don't have that many people in these studies. And throughout all of this, safety is the overriding concern. Nobody wants to make people sick. And so people who say the FDA is hiding things, they have data that show this is toxic or that is toxic, this is BS of the biggest order. It's a lie, it's misinformation. There's nothing hidden. I know all of these scientists, all they want to do is make something that is helpful for people. Okay, so I want to show you some kinds of cool screens that you can do, and if you're clever, you can develop others on your own. This is a mechanism-based screen. So we're looking for an inhibitor of a protease, okay? And we know the cleavage site of the protease. So we make a 4-amino acid peptide, and we put a bead on one side and something at the other end that's going to give off light of some kind that we can measure in an instrument, all right? And so when you mix the protease with this substrate, it's going to cleave it, and the light-producing material will be soluble, and the rest will be insoluble. And so you could centrifuge out the part with the bead and measure the light being produced. And that's shown on the right here. This is fluorescence intensity, so we made a fluorescence marker here with time, minutes. And you can see without the drug, very nice reaction of cleavage. And then you have one drug candidate here. It's going to inhibit the cleavage, so there's less light that is going to be produced because you're going to centrifuge out a lot of intact substrate, which you're not going to measure. So that's a good way to screen. And you can scale this into high throughput assays and screen lots of compounds. And we can also do this in cells. We can do it in bacteria, as shown here. Here's an assay where we're using tetracycline resistance. So tetracycline resistance comes about. Tetracycline is an antibiotic against bacteria. It's been around for ages. It comes about because a tetracycline-resistant bacteria has this multipass membrane protein in it that pumps out the tetracycline as soon as it gets in. And so the bacteria are resistant. So what you do is you engineer a HIV protease site into this multipass transmembrane protein. That's the green there. And then if you produce the protease in these bacteria, it will cleave this multipass protein, and the bacteria will no longer be resistant to tetracycline. Okay, so here's the cleavage, and this protein is now inactive because of that cleavage, and so the bacteria are inhibited by tetracycline. No colonies on a plate with tetracycline. But if you had a protease inhibitor, so the protease is this yellow guy, right, or gal, and if you had a protease inhibitor, it will block the cleavage, and this protein is now active, and you get colonies in the presence of tetracycline. So you can screen lots and lots of inhibitors just by counting colonies on a plate. So those are two very clever screens that you can use, many others as well, just to show you that you can do this very quickly. Now, in terms of the compounds, we have a kind of chemistry where, well, first of all, you could screen, if you had enough, you could screen up to 10,000 compounds a day, but even more. Companies have chemical libraries, as I told you, made for other purposes. It'll just screen them against your protein, your viral protein, natural products also. And we do combinatorial chemistry, where we have various linkers and fragments, chemical linkers and chemical fragments, and we combine them in different mixtures and make thousands and thousands of different compounds which could inhibit your target, and that's very common to do. You can also look at the structure of your target. If you have an enzyme and you know the structure, the active site, you can look at that and model something that might fit into the active site and make a compound that looks like that. Or you can even have the computer do the screening. You can say, here's the active site, and find something that'll fit into it. And of course, AI helps with that because you can teach it all the examples of what have worked, and then it learns that, and then it can predict new ones as well. And all this screening is done by robots. These are robots here. They're basically arms that take multi-well plates. So these plastic plates come in different sizes, 96, 384, and 1,536 wells. This one a human cannot work with. They're too small. You can't pipette it. It would take you hours. But the robot can easily pipette. You can pipette all the reaction mixtures into here. The robot will take the plate, put it in an incubator, time it, pull it out, put it in a reader, and all the information can be sent to you sitting at home having a cup of coffee, and you can analyze it. So many, many, many thousands of compounds a day can be screened using these robots. First question is, we have many antibiotics but fewer antivirals. What is a reason for the difference? Robotic screening is slow. There are few serious viral infections. Resistance is a problem. Antivirals must be potent. All of the above. How did we do here? Only 6 people are going to answer this question. Okay. How did the 6 people do? Antivirals must be potent. Robotic screening is not slow, and you didn't pick any of those. Resistance is a problem. Well, resistance is a problem, but that's not why we have so few antivirals, because you don't know about resistance until you make them, right? So that can't be it. It's number D. So let's talk about resistance. You will get resistance to any antiviral drug that you make, no matter how great you think it is, because viruses reproduce very efficiently and they have pretty good mutation frequencies, right? DNA viruses, intermediate, RNA viruses, very high. And if you give a person a drug for a long time, right, chronically infected people, HIV, HBV, HCV, you will start to select resistance because you're using a drug for long periods of times in the face of a lot of virus reproduction. And we have mutants to every one of those 100 antivirals that I showed you in the beginning. We have resistance to all of them, and that's kind of frustrating because we don't have a lot of them. Now that I think of it, actually, resistance to the SARS-CoV-2 antivirals has really not been detected, minimally. So the two drugs we're going to talk about, Paxlovid and remdesivir, I'm not aware of any resistance to those two. And so the only reason that would happen would be if the resistant viruses were less fit and they couldn't make it in the population. So maybe that's what's happening. So asterisk to that. So if you're treating a patient with an antiviral and resistance emerges, you can't continue it. So you have to monitor continuously, and then you have to use another drug. But sometimes there isn't another drug, and then the patient is in trouble. However, you should study the resistance, because that may show you, first of all, where the resistance arises and what gene or how, and maybe next time you can design antivirals that don't lead to resistance. So the mechanisms of drug resistance, of course, involve error-prone polymerases. As we've already discussed, and we will discuss again, the RNA viruses are error-prone, and they have no correction mechanisms with the exception of the coronaviruses and related viruses. So they have very high misincorporation rates, 10 to the 4 to 10 to the 5 nucleotides polymerase. That's 1 million times greater than the host DNA. And so in an RNA viral genome of 10,000 bases, that frequency of mutation leads to one mutation in between 1 and 10 genomes. And so in a cell, it's producing thousands of genomes. Every position can be, in principle, mutated. And this is the coronavirus exception. So coronaviruses here, Coronaviridae, that's the family. They are part of an order that has multiple families of related viruses. And these are called nidovirales. And they all have error correction exonucleases. So this is a picture of the RNA-dependent RNA polymerase synthesizing RNA. And there's one protein, NSP14, is an exonuclease that can detect an error, the wrong base put in, and will cut it out, and then the polymerase can fix it. So these RNA viruses have 10-fold less mutation frequencies than other viruses solely because of this. Now, DNA viruses, DNA polymerases also make errors, but DNA polymerases have associated with them error correction machinery. And so they can excise and replace misincorporated nucleotides. And so here's a picture of that. Here's a DNA polymerase copying a template, and a mistake is made, this little mismatch here. You can see they're not base paired. There's an exonuclease associated with the polymerase that will chew out, remove that mismatch, and then the polymerase will recopy it. So the error rate is slower than RNA viruses, and so DNA viruses evolve more slowly than RNA viruses. They simply have less diversity, which is not to say you don't get the resistance of DNA viruses to antivirals, it's just slower. All right, so here's, again, the virus reproduction cycle, all the different steps that we've learned about in the first half of this course, and we have inhibitors to every one of them. I want to go through some of these and talk about how the antivirals work. And let's start with amantadine, the influenza inhibitor that we don't use anymore because we have widespread resistance. But it's a very, very cool mechanism. And remember, this drug was discovered in the 60s, and we used it for a long time before we understood how it worked. So the drug actually interacts with the viral M2 protein, which is an ion channel. So here's the influenza virus particle with its membrane. And aside from the hemagglutinin and neuraminidase, there's another membrane protein, which is an ion channel called M2. Now, M2 normally functions during virus entry. When the influenza virus is taken up into the endosome. The endosome acidifies as it moves towards the nucleus. And the protons that are in the endosome, right, that's how you lower the pH, you bring protons in. Those protons go through the M2 ion channel into the virion interior. All right, so they go through the M2 channel into the interior, and then when membrane fusion occurs, that allows the ribonucleoprotein to come out. If the interior of the particle had not been acidified, the RNP would never come out and infection would be blocked. So that's how amantadine works, by blocking acidification of the particle. Now don't get it confused with acidification of the endosome. That's different. The endosome is outside of the particle. And that acidification is needed for membrane fusion. But unless the particle interior is acidified, the RNA will not get out. So amantadine prevents uncoating of the virus particle. And here's a structure of the M2 ion channel. So here's the influenza virus membrane, and the M2 ion channel is a very short protein, and the active channel is a tetramer, 4 chains, as you can see here. And the protons will simply flow through from the endosome into the interior of the particle. It's a flow. It's not a pump, it's a channel. The protons simply flow through. And when amantadine is present, it will block the flow of protons. Amantadine can bind in the channel, as shown. This is the amantadine in red here. It can bind in the channel and block the proton flow, but it can also bind outside of the channel and cause a conformational change in the M2 protein that blocks flow of protons as well. Right? And so it's easy to get resistance to amantadine. You simply have amino acid changes in the M2 protein that prevent it from binding. So that was an entry. That was a entry inhibitor. Here's another entry inhibitor for HIV. It's called Maraviroc, and it's a CCR5 inhibitor. So remember, the HIV shown here has a spike glycoprotein that binds first CD4 and then CCR5. Interaction with both receptors is needed for infection. Maraviroc is a small molecule. Here's Maraviroc right there. And that's the molecule, and then on the right is Maraviroc with, CCR5 in orange with Maraviroc bound. You can see Maraviroc in blue. So Maraviroc, small molecule that binds CCR5 and prevents the spike from binding to it. So it inhibits entry. This is still used. It's quite useful, but only in combination. If you use it on its own, you're going to get resistance. And you may ask, why isn't this toxic? Why isn't you're inhibiting CCR5 basically? Why isn't it toxic? Well, do you remember that 16% of humans have a deletion of CCR5 gene? So obviously it's not essential, so you can inhibit it. Okay, polymerase inhibitors, this is a big one. So what we do to make polymerase inhibitors is there are 4 bases you need to make nucleic acid, right? A, G, C, and T or U, depending on if it's a DNA or RNA virus. And then you modify them chemically to get all these drugs on the outside that are potential inhibitors of nucleic acid synthesis. And many of these are nucleoside inhibitors, okay? And we're going to look at acyclovir, which is a great anti-herpes simplex virus drug. It's a nucleoside analog. Now, catch this. This is very important. Nucleoside means there's no phosphates on this. And phosphates for many years, if you had a phosphate on your molecule, it makes it hard for it to get into a cell. We fixed that problem later. I'll tell you how. But if you put phosphates on, they're called nucleotide analogs. So as soon as you put the phosphate on, it gets a T instead of an S. All right? Remember that. Okay, so acyclovir is a nucleoside analog. So you can see here, acyclovir is made by modifying guanosine, by basically wrecking the ribose. as you can see here. And this hydroxyl, of course, is going to be needed for adding the next base. So it's not there in acyclovir, so you interrupt nucleic acid synthesis. Let's take a look at how that happens. Now, acyclovir is very cool because, well, it needs to be phosphorylated, right? So here's acyclovir. It gets into the cell. It will only be phosphorylated by a herpes simplex virus protein called thymidine kinase. It adds 1 phosphate. It will not be initially phosphorylated by any cell kinase. So that's great because it doesn't have any toxicity in uninfected cells. Amazing. Once that 1 phosphate is on, then the cell kinase is put on the second and the third. These are cell kinases, GMP and NDP. And now it can be used by the polymerase. You know, you need 3 phosphates on these nucleosides in order to get them into DNA, right? So you have to put 3 phosphates on any drug that you make that's targeting nucleic acid synthesis. And again, remember, it's a derivative of guanosine, but it's very different. So the viral DNA polymerase will incorporate that into the DNA, and that's it. Can't incorporate any more because there's no hydroxyl. You need a hydroxyl to add the next base, and there is none on acyclovir. So that's the mechanism of inhibition. Now, the original acyclovir was not terribly bioavailable after oral administration. So you can put acyclovir on herpes simplex cold sores, it's fine, but if you need to take it orally, which is convenient, especially for certain medical conditions, you need to modify. And this is a cool way that they modified acyclovir. They added an amino acid to it. So here's acyclovir on the bottom. They added valine. So this drug is now called valacyclovir. You can take this orally. It gets into cells, and then the cell takes off the valine, so you have acyclovir in the cell. So it's orally bioavailable, and it is now in the cell as acyclovir. So that's what we use now instead of acyclovir. Now, acyclovir resistance arises during virus replication. And there are 2 kinds of mutants. So remember, HSV-TK has to phosphorylate acyclovir initially, and some of the resistance are in the thymidine kinase gene, and they cannot phosphorylate the drug, so the virus is resistant to it because it never gets phosphorylated, it will never get incorporated into DNA. And then there are another class of mutants in the viral DNA polymerase gene itself, so that the polymerase cannot recognize acyclovir, even though it's triphosphorylated, will not incorporate it, and so again, there's resistance. So 2 ways you can get acyclovir resistance in herpes simplex virus. All right, so another interesting chain terminator is AZT, azidodeoxythymidine. This is the first approved HIV drug. This is a drug that had already been known, and it was being checked out to see if it would be a good anti-cancer compound. And you know, early in the AIDS pandemic, people said we need to try everything. So they screened and they found out that this inhibited HIV replication. So here's AZT. Let's show you, well, you don't even have to see what the parent is. You can see it's got a ribose, but instead of a hydroxyl, it's got nitrogen down there, an azido group. Why it's called azidodeoxythymidine. You cannot add another base if you've got nitrogen there. You need a hydroxyl. Now this drug gets into the cell and is mono, di, and triphosphorylated by cellular kinases. There's no specificity as for herpes simplex because there's no viral enzyme that phosphorylates this. This is then incorporated by the reverse transcriptase, and it stops chain synthesis because of the azido group there. You can't add another base. Now, it turns out that it's a better substrate for HIV RT than for cellular polymerases. So there's a little bit of selectivity, but it has lots and lots of side effects because It also can inhibit cellular DNA polymerases, but it was the first thing we had, and it was the only thing we had. So substantial side effects, unlike acyclovir, right? It's given orally, and the half-life is 1 hour. Remember I told you before, it's crazy to have a drug with a 1 hour half-life. Well, this is the only thing that was available. And you know, the people who had AIDS were very active, and they said, we want this. They went to the FDA and said, we want this. So it was approved, and they had to dose 2 to 3 times a day. And within a year, we got resistance as a consequence. But it did save some lives initially. And that drug is the story of Dallas Buyers Club, that movie, how it was hard to get it, and patients found they could break it in parts. You didn't need to take the whole thing, and it would still work. And that allowed the FDA to reduce the dose and prolong the supply, basically. And resistance to AZT arose right away. Single amino acid change at one of 4 sites in the reverse transcriptase, and the altered RTs do not bind the phosphorylated AZT. That's the mechanism of resistance. So people said, okay, we're going to beat this, and they develop other nucleoside analogs. Nucleoside, no phosphates, right? Diadenosine, zalcitabine, stavudine, lamivudine. And they were then, since we got more than one, they said, let's use them in combination. So they used 2 at a time. And you delay resistance, but you still got resistance in less than a year when you use drugs in pairs like this. But that's the story of the nucleoside inhibitors for HIV. Now, they're also developed subsequently non-nucleotide inhibitors. Okay, these are drugs that bind away from the active site. They're not a chain terminator. They bind away from the active site. There's the red spheres there. That's one of these drugs. They change the conformation of the polymerase a little so it doesn't work anymore, right? So they're non-nucleoside inhibitors. They're not incorporated into the chain. And so we have nevirapine, delaverdine, and efavirenz, 3 different ones that have been developed. When used singly, you get resistance immediately. And there are amino acid substitutions in sites that are around the drug, the binding site of the drug. So you can't use these alone for treatment of AIDS. Now, today we still use them, but in combination therapy. That's the lesson that was learned from all this. And I'll come back to that in a bit. SARS-CoV-2, we have nucleoside analogs. We have remdesivir, which was the first to be developed. This is a prodrug, which again means it has to be phosphorylated to be monitored. So here's it's a prodrug of adenosine. It's shown right here. This is adenosine at the bottom, and this is remdesivir. All right. So you can just see the adenosine in here, right, on the right. But then it has all this other stuff, which is very interesting because notice there's a phosphate in there. The chemists figured out how to bury the phosphate, look, among all this other stuff. You see, I'm not a chemist because I call it stuff. But these other side groups bury the phosphate. So now it can get into cells, and in the cell actually takes off everything, and I believe it leaves on 1 phosphate, so it can then be di- and triphosphorylated, and it works. So this is the solution to the phosphate problem. The reason this is important is because putting that first phosphate on a drug is really slow. The next 2 are fast, but the first one is slow. So if you can already put a drug in with a phosphate, it's so much better. Anyway, remdesivir was developed for Ebola virus. It turns out to inhibit multiple RNA viruses by chain termination. It was found to inhibit SARS-CoV-2 replication in cells, and it was licensed in the US after phase 3 trials. We went through all the trials. Has to be given intravenously. Okay, that's the problem. So you either have to go to a hospital or an infusion center, and it has to be given early, within 7 days of symptom onset. If you do that, you have an 87% reduction in progression to hospitalization and severe disease. Now, You note here I say no effect on hospitalized COVID patients. Actually now they have done some clinical trials and there is an effect even in a hospitalized patient when it's very late and virus reproduction is low. Remdesivir can have a positive effect. All right, so that's a nucleoside analog for SARS-CoV-2. Maybe it should be called a nucleotide analog because it has a phosphate. I don't know, it's a good question. Maybe it's not in the right, because it's got all that other stuff around it. I have to look into that. Then we have another one, molnupiravir, which is a prodrug of cytidine nucleoside. So there's cytidine. And then we have the final drug. This is an intermediate, and this is the final drug where you can see that they've added side groups here where the hydroxyl normally is. And that's the only other chain. So this one is a mutagen. It doesn't chain terminate. It templates as U. So it's a C. So it's put in whenever there's a G. But then the polymerase sees it as a U. And so it mutagenizes the genome because you make base changes. And so this was given licensure in the US. This is orally bioavailable, which is great, but only 30% reduction of progression to hospitalization and death. Not the greatest, but for some people it's the only option because remdesivir is not working for them. Okay, those are some examples of inhibitors of nucleic acid synthesis. Resistance to which antiviral would involve amino acid changes in a viral enzyme? Acyclovir, amantadine, penicillin, all of the above. So I don't know why it didn't register, but the answer is acyclovir. Amino acid changes in a viral enzyme. It's either thymidine kinase or the DNA polymerase of herpes that sustains amino acid changes to make resistance to acyclovir. The amantadine resistance is in the M2 channel. It's not an enzyme. And of course, penicillin is not an antiviral. All right, we have integrase inhibitors. So this is a step unique to retroviruses, right? The genome is reverse transcribed to DNA. It integrates. The integration reaction is carried out by integrase, which we've looked at this before, and it's repeated here at the bottom. The integrase molecule, which is a tetramer, takes retroviral DNA and links it to host DNA. So we have a couple of integrase inhibitors, raltegravir, and I don't remember what DTG is. But the way they work is shown here. So this is a very close-up view of the active site in the enzyme, in the integrase. So here we have the 3' end of the viral DNA, and here is the active site, 2 aspartates. Remember, polymerases have 2 aspartates to chelate magnesium. The same thing with integrase. And so this is getting ready to link the 3' hydroxyl to cellular DNA. But when you put drug in, raltegravir or DTG, it pushes the 3' end away from the asps, and this blocks them too. So you see the 3' end is right near the 2 asps here, and with the 2 drugs in, this way here, the DNA is pushed towards us, and here, same thing. So in both cases, it pushes the DNA. So now there's no chance that this can be linked up to host DNA. That's how these inhibitors work. I don't know why this is here. It's a polymerase inhibitor. Bless you. Okay, it doesn't matter. So this is a RNA polymerase inhibitor for hepatitis C virus. And it's called sofosbuvir. It's a prodrug also. So it's a nucleoside analog. And you can see, well, here's a ribose here. Here's a base. And it's got fluorides here, and then it's got this other group here with the phosphate buried. So again, the same strategy as used for remdesivir, to bury the phosphate so that it gets into cells. So here it is in the cell. Normally, the first phosphorylation reaction would be slow, right? But here we're putting it in with 1 phosphate already, so cellular enzymes could add this second and third phosphate, and now it's active as an inhibitor. It's a chain terminator. It costs $1,000 per pill. 12-week treatment, $84,000. But it will get rid of hep C. Okay, now this is a relatively new influenza antiviral, which was approved in 2018. It is an inhibitor of the endonuclease. This is very cool. So remember, the influenza polymerase requires primers for mRNA synthesis, and these primers are derived by cleavage of host cell mRNAs. You get these 13 base primers that then initiate mRNA synthesis. And the endonuclease that cleaves this is a viral protein. So people took that protein and did screens for inhibitors, and they came up with this molecule, which inhibits it. And is a very good antiviral, actually. It's actually better than the neuraminidase antivirals, which we're going to talk about in a moment. Protease inhibitors are a big target. Many viruses have proteases. For example, retrovirus, HIV protease is absolutely required for the production of infectious virus particles. After the particle buds, the protease in the particle cleaves all of the precursors to make the final viral particle that's infectious. And we've talked about this in the assembly lecture, so we don't need to go into it here. So there are multiple protease inhibitors. Here's one called ritonavir. And what they did to develop ritonavir is they said, okay, what is the substrate for the protease? It should be protease substrate. And they found the amino acids where it was cleaved. They modeled the transition state before cleavage occurs, and they came up with this drug that would mimic the transition state. And then they modified it further and came up with the final drug, which is ritonavir, which is basically a peptidomimetic. It looks like the cleavage site. It looks like all the amino acids of the cleavage site, but it has features that prevent it from being cleaved. It goes into the active site. Here's the protease, and there's ritonavir in red, bound in the active site. It binds in there. The protease thinks it's a substrate. It binds in there and stays there and doesn't come out, so the protease can't cleave viral proteins. So these are called peptidomimetics. We also have the same for hepatitis C virus. The hep C genome, remember, we have a polymerase inhibitor already, but we also have peptidomimetic inhibitors. The genome is a long protein that's cleaved by proteases, and one of the main proteases is NS3. Cleaves all these. This is essential for replication. So a drug, a peptidomimetic, was designed in a similar way to leprevir that binds in the active site. You can see it in red here of the protease and completely inhibits it. Now on their own, you get resistance to these, but you have to use them in combination therapies. We have a protease inhibitor for SARS-CoV-2. It's called Paxlovid. Paxlovid is actually two drugs. It is a protease inhibitor and also another drug that prolongs its half-life in cells. So the coronaviruses also make a polyprotein that has to be processed by the main protease called Mpro. And so peptidomimetics were designed. Here's Paxlovid, or the inhibitor component. What is it called? Nirmatrelvir, that's the protease inhibitor. Paxlovid is the combination of two drugs. Nirmatrelvir and ritonavir, actually. So ritonavir, which is the HIV drug, turns out to inhibit an enzyme in your liver called cytochrome P450, which turns drugs over. And so it makes drug half-lives longer. It's used in other drug formulations as well. Anyway, this is nirmatrelvir, the protease inhibitor. It binds in the active site of the protease and is very effective at inhibiting SARS-CoV-2. You can see the black line here. At nanometer concentrations to inhibit 50%, but it also inhibits some of the other coronaviruses, including SARS-CoV-1 and the common cold coronaviruses and MERS. And you might modify it just to get them that drug more active against these other viruses. So that was licensed for the inhibition of SARS-CoV-2. So I had COVID once and I took Paxlovid, and in one day all the symptoms were gone. It's really great. And I went to a meeting in 2022, got COVID, took Paxlovid, was gone. Except the only problem is you have to take it for 5 days, and it gives you this horrible taste in your mouth for 5 days, but it's better than getting COVID. All right, so for influenza virus, we also have neuraminidase inhibitors. Remember, the neuraminidase works as the viruses are budding from the cell. There's receptors on the cell, so the viruses would be binding to those receptors. So to get around that, the viruses have an enzyme called neuraminidase, which cleaves off the receptors from the host cell so the viruses can leave. So people thought, well, why don't we make inhibitors of the neuraminidase, and that will keep the virus from leaving the cell. And in fact, that's how they work. There are two inhibitors. Here's sialic acid, which is the receptor for the hemagglutinin. And sialic acid also binds in the neuraminidase. This structure here in B is the neuraminidase, and the sialic acid binds in it. So this is actually a computer-designed drug. They said, make something that looks just like sialic acid. They came up with two drugs, Relenza or zanamivir. This is inhaled, so not many people take it. And then Tamiflu, which is the oral medication that you may be more familiar with. They bind in the active site and prevent the neuraminidase from binding to the sialic acid. Now, they're designed to mimic sialic acid, and the idea was if you make the inhibitor really close to sialic acid structurally, you can't get resistance because then the virus can't bind sialic acid. And so that seems to have worked. So here's the diagram of the inhibitors. We have sialic acid binding to neuraminidase, right? And then we have the two inhibitors, also Tamiflu and zanamivir, or Relenza. Relenza looks closer to sialic acid than does oseltamivir. That's why I've drawn it differently here. And so it's relatively easy for changes in the neuraminidase to be selected for that make for oseltamivir resistance. It's harder for zanamivir because it looks very much like sialic acid, and if you change it to evade zanamivir, then you can't bind sialic acid and you won't be able to replicate. So now we get the numbers from the previous one. Okay, so it's going to work. All right, which of the following targets for HIV antivirals inhibits the earliest stage of infection? Nucleoside inhibitors, NNRTIs, CCR5 inhibitors, integrase inhibitors, fusion inhibitors. Earliest step of infection. So we're going to wait till the end of the lecture, then we'll see the numbers. But the right question is CCR5 inhibitors, right? They're inhibiting binding, which is the earliest step. Can you make broad spectrum antivirals? This one, favipiravir, is a broad spectrum inhibitor of RNA viruses. It is a nucleoside analog, so there it is. So here it is outside the cell. It's really very different from the bases that we've seen. It just has the base. There's no ribose. So it gets in the cell, and then a cellular enzyme, HGPRT, has to add a ribose, and then it's triphosphorylated in the cell. So this inhibits a whole range of plus and minus strand RNA viruses. It's licensed in Japan to treat influenza, but it is not licensed here in the US. And I'm not sure why. Maybe it's not potent enough, or maybe there are other influenza inhibitors already. So, but the principle is that you could hit all RNA viruses. And the same for DNA viruses. Cidofovir is a broad spectrum inhibitor of DNA viruses. It hits all those DNA viruses. And again, this is an acyclic cytosine phosphonate. Remember, there's the solution to hiding the phosphate. You can see the ribose is gone, the base is there, and they've hidden the phosphate in some oxygens and hydroxyls. And it's a mimic of deoxycytidine monophosphate. As a result, it's diphosphorylated, so that bypasses that first phosphorylation step, which is very slow. And the diphosphorylated form has a higher affinity for viral polymerases than the host, which seems to be a property of these acyclic non-nucleotide analogs. So again, this inhibits a lot, but it's not been licensed for anything in the US. I want to end up by talking about two stories of antiviral success: combination therapy for AIDS and Hep C. So we know the replication cycles of HIV and hepatitis C virus, and we have many inhibitors at different steps of both virus reproduction cycles. And these are lifelong infections, and so it was obvious that you could develop inhibitors. You had a lot of time to develop them, and you have a lot of time to prescribe them to people. So for HIV, we use combination therapy, highly active antiretroviral therapy. We have a pill that combines 3 or 2 different inhibitors, and it doesn't cure your infection. You still have DNA integrated into long-lived cells, but you can live your life having this and not have all the symptoms of AIDS. So here's the math for drug resistance. If you assume 1 amino acid change or 1 mutation is needed for drug resistance, the mutation rate of the virus is 1 in 10 to the 4 bases polymerized. So each base is substituted in every 10,000 viruses, each base in the genome. Infected people make 10 to the 10th new viruses a day. So 10 to the 6th viruses will produce each day with resistance to 1 drug. That's why 1 drug doesn't work, okay? What about 2 drugs? They're developing resistance now, 10 to the 4 times 10 to the 4. 100 viruses resistant to 2 drugs arise each day, which is better, but you still get resistance within a year. And then 3 drugs, you need 10 to the 12 viruses, which is very hard to achieve, but it does happen. People develop resistance to 3 drugs, and so now, and they circulate these viruses. So now if you are diagnosed with HIV infection, they take your virus and sequence it to see if it's resistant to any triple combination. And they give you the right combination, because there's no point in giving you a drug that's not going to work. So these are all the antivirals against HIV, the different targets. And down here, the combination therapies, 2 or 3 combination. 2 have very specific uses, but the combination 3, like 2 is used in PrEP, for example. That's sufficient for PrEP. But 3 are given to people with established infections. And these save lives. This is the actual and projected number of people receiving antiretroviral therapy from the years 2003 to 2018, in different regions, different WHO regions. And Africa has the biggest burden of AIDS, as you will see, and they're getting the most antiretrovirals. You know, the USAID program funded a lot of this, and this has been stopped. So this is going to go down again, unfortunately. And then at the bottom, HIV/AIDS-related deaths due to antiretroviral therapy. 10 million total adult deaths averted. And here is pregnant mother to child transmission. With combination therapy, these are the deaths that have been averted. So these work very well. We also have PrEP, pre-exposure versus post-exposure. This is daily double therapy, tenofovir and emtricitabine. For those who are at high risk for infection, it reduces the risk of sexual transmission by 90%, and it reduces the risk of transmission by intravenous drug use by 70%. And then Hep C has also responded. In this case, you can cure Hep C infection. So this is a history of antiviral treatment of Hep C. So these are the weeks of treatment, and the orange is the longest treatment, and the blue is the shortest. So back in 2013, we used to treat patients with interferon and ribavirin, and it sometimes required 80 weeks, but didn't always work. And then we added an inhibitor, boceprevir and telaprevir, and then that shortened it a bit. But then in 2015, we started using combination therapies. That reduced the time even more. And then finally now we can reduce the treatment to 20 weeks or less and achieve complete cure of infection. So really the Hep C story is remarkable, except many people who get Hep C, they take drugs, they cure it, and then they get it again because they're intravenous drug users or something else which predisposes them to getting it over and over. So we still need a vaccine for Hep C, and we don't have that yet. However, as I told you on day one, there are 10 to the 16th HIV genomes on the planet today. So that means we have resistance to all the antivirals that we make and will ever make. So we always have to be alert to screening people to see what resistance they have and give them the appropriate therapy. All right, next time we're going to talk about evolution, how viruses change over time.